responding. No, I hope others are. I hope they are online. Is that Melissa Akinleye?
Yes, sir.
Yes. Okay. Anyway, when we get to the day, we shall cross it. Let us know who I'm working with, so that we know how we are going to define the project. Yeah, let's start. What we are going to do today is not something that's going to drag us like that. We just want to do history of pathogenic um, microbiology. I do not have any slide to present today, but we'll talk about some things maybe later. I can share my lecture notes, but today I'm not going to share it. I just want us to do the lecture today because we have been postponing. Let's just do it today so that we know we have started. Are we on the same page? Hello? Hello?
Hello, sir.
Are we on the same page? I can't hear anybody.
Yes, sir.
A lot of people have muted themselves, or what is happening?
We are here, sir.
I can see a lot of people have muted themselves.
We are with you, sir.
All right. Okay, let's quickly go through the history of pathogenic microbiology. And what do we understand by pathogenic? Pathogenic means that organism that is able to cause disease in a host. And these organisms, they can be bacteria, virus, parasites, protozoan. We have a whole lot of them, but majorly we'll be speaking to bacteria, viruses, and parasites. Are we there?
Yes, sir.
Yes, sir.
Okay. So the word microbiology, what does it mean? It is derived from three Greek words. The microbiology we are talking about, because it's from microbiology that we have microorganisms. So the microbiology we are talking about is from three words, Greek words. The first is micros, meaning small. The second one is bios, that is BIOS, meaning life, that is small life. And the third one is logia, Logia, meaning science or study. That is the study or science of small life. That's the meaning of microbiology. So, like I did say the other time, microorganisms includes bacteria, fungi, protozoa, algae. It also includes viruses. So, discovery of microorganisms. How did they discover microorganisms? Before microorganism was discovered, microscope has been discovered, because microscope was the only instrument that was able to magnify microorganisms to unheaded high. Are we there? Are we there?
Yes, sir.
Yes, sir.
Okay. So the invention of microscope enabled the study of microorganisms. That's what I just explained. Without microscope, you won't be able to see microorganisms. So before you can say you identify any microorganism, microscope has been invented. So, and the first microscope, the first ever microscope was simple. It was simple microscope. It has magnifying lenses that magnify the image of the tiny life. Those people that first observed the previously unseen microbial world were Robert Hooke and Anthony van Leeuwenhoek. I think we are very used to Robert Hooke, but let me spell that Anthony van Leeuwenhoek. Robert Hooke is Robert. Hooke, Hooke, Robert Hooke. And there were two individuals, and Anthony Van, Vean, like Van. Leeuwenhoek, Leeuwenhock. So those are the two individuals that were able to observe microorganisms using microscope. And microscope, like I told you, it has magnifying lenses that magnifying the image. So let us go and look at the works of all these earliest microbiologists. Robert Hooke, 1635 to 1703. Robert Hooke was able to live between 1635 to 1703. Robert Hooke is an English mathematician and natural historian, and is the first to coin the term cells. All these cells that we are using today was the first person to use that term, cells. So Robert Hooke was able to make a known description of microorganisms. Then the second man that we call Anthony van Leeuwenhoek, that one lived between 1632 to 1723. This one was a Dutch. When they say somebody is a Dutch, that means that it's from Netherlands. And he was an amateur microscope builder. Just developed interest in building microscopes. And that's why I said that the first microorganism was observed. Microscope has been invented. And the man that invented the microscope is this Anthony van Leeuwenhoek. Anthony van Leeuwenhoek was the man that invented microscope. So the microscope that he invented that time was capable of magnifying an image about 300 times. That is, an image, it can magnify it 300 times. So
His slide is not
this
visible.
man, what happened?
His slide is not visible, sir.
I am not sharing any slide today.
Okay, sir. Thank you, sir.
So I'm not sharing slide. I will give you note later. I just want us to understand something. I'm not sharing slide. So just listen, and anything you can jot it down. So this man called Anthony van Leeuwenhoek observed microorganisms, and he called them animalcules. Animalcules. He called the microorganism that he saw that time animalcules. The same man, Anthony van Leeuwenhoek, discovered bacteria in 1676. Anthony van Leeuwenhoek published extensive and accurate observations of microorganisms. So this man is regarded as father of microbiology. Now, let's leave Robert Hooke and Anthony van Leeuwenhoek. These are the earliest microbiologists that let us know what is microscope, especially Anthony van Leeuwenhoek. And he named the microorganism animalcules. Now, after this one, there's what we call the spontaneous generation conflict. Spontaneous generation conflict. What is the spontaneous generation conflict? It's talking about that living organism develop from non-living organism. That is what spontaneous. That's what spontaneous generation was talking about. And why they say there was conflict is that some people agree to that, while some people oppose it. There's nothing like that. That living organism couldn't have developed from non-living organism. Are we there?
Yes, sir.
Yes, sir.
What did I call spontaneous generation conflict?
Hello,
If
sir.
you are following me, what did I call spontaneous generation conflict?
Hello,
You
sir.
said living organism
Living
developing
organism
from non-living.
develop on non-living
Yes.
organism.
And why was there conflict? I say
Because
some
some
of
people
them,
believe
some of them agree to that, while some people say no, it couldn't have been. But the people that oppose it, they carry out experiment to oppose that spontaneous generation. So that concept, the concept of spontaneous generation is that living things, so we are developed from non-living things. That is like decomposing matter. That's what the spontaneous generation is all about. But some people say no, it's not possible. the living things or the living organism couldn't have developed from decomposing matter, which is not living thing. For example, they were telling us that time is that if you put your food on the ground or on the floor, before you know it, you will see maggot there. Maggot is seen as a living organism that developed from non-living organism. Are you there?
Yes, sir.
So now some group of microbiologists now say that maggot is not from the organic matter that is there, the decomposing organic matter. It's not from the food, or it's not from the meat. It's not from anything. That it is because flies were able to get access to that food that they were able to deposit their eggs, and the eggs hatch into larva, which is called maggot. So the maggot is not from the food, it's from the external source. That is from the ants that perch on the meat or any putrefying material that it is not. That is, if you did not allow flies to get very close to that meat, there won't be maggot on it. And they did the experiment. They covered the meat. They made sure that fly did not get very close to it. And there was no maggot. And the one that they left open, that flies were able to get in contact with, there were maggots. Do you get my explanation?
Yes, sir.
So you will be able to explain spontaneous generation conflict. Are you there?
Yes, sir.
Yes, sir.
Okay. We can't take all these microbiologists one by one because there are many. I will just try to pick those that their names we are familiar with. I've talked about Robert Cook, Robert Hooke. I've talked about Anthony van Leeuwenhoek, rather. Then let me go to the work of Louis Pasteur. At least we know Pasteur Pipette now. Hello?
Hello.
I say we all know Pasteur Pipette.
Yes.
So the man that Pipette was named after is Louis Pasteur. And he lived between 1822 to 1895. Louis Pasteur was a professor of chemistry, and he devised series of neck flasks. So what did Pasteur did? Louis Pasteur, in order to disprove that spontaneous generation concept, he now devised his own flask. So he had two flasks. He left one without cotton wool so that air can enter and air can come in. And the other one, he plugged it with sterile cotton wool. It was discovered that the one that he plugged with sterile cotton wool, the meat that was put inside was not contaminated. There was no growth, there was nothing there. The thing still remained like that. But the one that that have no cotton wool, there was free passage of air into the flask. I would discover that one was contaminated. So news was telling them that whatever that might have been growing on the meat is external. It's not from that meat. He was able to do that experience and everybody believed. Are we together?
Yes, sir.
So Louis Pasteur was able to defeat the concept of spontaneous generation with his experiment. So another work of Louis Pasteur is effective sterilization method, which involves holding juice and milk at 62.8 degrees Celsius for 30 minutes, and that is called pasteurization. So pasteurization is the work of Louis Pasteur. Louis Pasteur went further again to discover alcoholic fermentation that was catalyzed by yeast cells. He also developed vaccines for disease anthrax, fowl cholera, and rabies between 1880 and 1890. Louis Pasteur was able to postulate germ theory of diseases. And what does germ theory of disease state? The germ theory says that there is always a specific microorganism that cause a specific disease or disease. For example, it was able to show that Bacillus anthracis was the etiology of anthrax disease. That is, for any pathogenic disease, there is specific microorganism causing it. And that is what is called, what did I call it? What did I call that theory?
Germ theory of diseases.
Germ theory of disease. That is from Louis Pasteur. So, and what is can you tell me in one word what is meant by germ theory of disease?
There is always a bacteria responsible for a particular disease.
Yes. It may not be bacteria, but there is always
Organism.
a specific organism responsible for a particular microbial disease. So this Pasteur usher in golden age of microbiology. So you could see this Pasteur did a lot of work on microbiology. Now, after this Pasteur told us that there is a specific organism causing a specific disease, other people work on that. And that led us to recognition of the role of microorganisms in disease. So there are many of them that work on all this role of microorganisms in disease. But let us pick the one that is popular that we are used to. Let us use Robert Koch. Robert Koch lived. Robert Koch lived between 1843 to 1910. Robert Koch was a German physician. And he was first to directly prove that the role of microorganisms in causing disease. He established relationship between Bacillus anthracis. So what I credited to this Pasteur, it was actually Robert Koch that had that credit. He was able to prove that Bacillus anthracis, that organism was the one responsible for anthrax disease. That is Robert Koch, not Louis Pasteur. So, what are the works of Koch? So they call it Koch postulate. How did Koch was able to establish that Bacillus anthracis was the etiology of anthrax disease? Was that he was able to inject a healthy mouse with blood from a diseased mouse, and the healthy mouse quickly developed anthrax. That is, some mouse that is sick, they collected his blood and injected it with the one that is not sick, and that mouse that is not sick developed sickness. So they were able to know that this anthrax was caused by Bacillus anthracis. So let us mention some of the Koch's postulates. That is what Robert Koch postulated. There are so many of them, but let us just pick one or two. One of the Robert Koch's postulates is the suspected disease-causing organism should be present in all cases of disease and absent from healthy animals. That's what we have explained the other time. That for every given microbial disease, there must be specific microorganism causing it. That's what he was trying to explain here. Robert Koch was also the first to grow bacteria on solid culture media. This is one of also his discovery. Robert Koch was the first person to grow microorganism on solid culture media. We have so many things that Robert Koch did, but let us leave it at that and go to the work of Edward Jenner. Edward Jenner. Jenner is Jenner. So, what did Jenner do? Edward Jenner was able to use fascination procedure to protect individuals from smallpox. What that means that Edward Jenner is the father of fascination. He was the first person that introduced fascination. He was able to protect individuals from smallpox using fascination. Are we following?
Yes, sir.
So, because all this one that I am mentioning, it can come up in objective. It should be able to tell us the work of each of these microbiologists. What are they known for? So, let's leave because there are so many of them. Just let us just not bother ourselves with so many names, because if we continue like this, we will even 3 hours we have not finished this lecture today. So let us just leave it. If I've been able to bring out some names that we are familiar with, and what I was able to add that I can call value addition was that I was able to highlight their work so that anytime they mention Robert Koch, you know what is known for. Anytime they mention Edward Jenner, you know what Edward Jenner was able to do. So there are a lot of inventions, but let us just go to era of molecular microbiology. The molecular micro, Are you calling me back? Hello?
No, sir. Go ahead, sir.
Okay. The molecular microbiology began in the 1950s. Advancement in the knowledge of bacterial physiology, biochemistry, and genetics. Genetic manipulation, which involve transfer of DNA from one organism into bacterium and protein encoded by DNA, have first led to the development of the field of biotechnology. So you can see there's no way you talk about molecular microbiology without talking about DNA. Take DNA from one organism and transfer it to another organism. Whatever that is encoded in that DNA automatically is transferred to the other organism that the DNA is transferred to. So that's what molecular microbiology is all about. Then this one led us to DNA sequencing. This one refers the phylogenetic, that is the evolutionary relationship among bacteria. Some bacteria are more related to others, while some are not related. So that is what they call phylogenetic relationship. So, which led to revolutionary new concept of microbial systematic. In the 1990s, DNA sequences gave back to field of genomics. So what we are talking about now is genomics. So we have scope of microbiology. We have so many. We have the basic and we have the applied scope. Under the basic, okay, the two of them, the two branches are intertwined and are complementary to each other. The basic branch of microbiology is concerned with the study of biology of microorganisms. Field of study includes, under the basic one, is bacteriology is one of them, mycology, another one. We have protozoology, we have microbial cytology, we have microbial physiology, microbial genetics, microbial ecology, microbial taxonomy, virology, medical microbiology, agricultural microbiology, industrial microbiology, aquatic, all these agricultural, industrial, aquatic, they are all applied microbiology. We have public health microbiology, we have medical microbiology, all these ones that we have been mentioning, yeah. Applied microbiology, starting from medical microbiology, agricultural microbiology, industrial microbiology, aquatic, public health, immunology, food and dairy microbiology. We have endless list like microbiology. Now, let us take bacteria. The bacteria domain, because we are going to be talking about this gram-positive bacteria and gram-negative, so that's why I want us to have some knowledge of bacteria. so that by the time we are picking them one by one, at least you know. Bacteria are prokaryotic, that is, they lack well-defined nuclei. That is plural of nucleus. So when they say an organism is prokaryotic, the meaning is that they lack well-defined nucleus and membrane-bound organelles. Bacteria comes in many shapes, or bacteria come in many shapes and sizes, from minute spheres, cylinders, spiral threads, to flagellated rods and filamentous chains. What are the structure of a bacterial cell? Capsule is one of them, though not all of them have capsule. Not all of them are capsulated, but we have capsulated bacteria. Capsule is made up of polysaccharide, that is complex carbohydrate. What is the role of capsule? The role of capsule is to keep the bacterium from drying out and to protect it from phagocytosis. What is the role of capsule? Hello?
It's
What
for
is the role of
protection, sir.
protection? In what way? Speak like a microbiologist now. There are some words that you
Bacteria from drying off
write.
and to prevent phagocytosis.
Exactly. You need to speak like a microbiology, not that it's for protection. Protection from what? From so let us know what you are what is protecting. Phagocytosis. If you have not mentioned phagocytosis, you have not gotten anything. If you like, write one page on capsule. If you have not told us what it does, that it protects bacteria from phagocytosis. What is the meaning of phagocytosis? Engulfing. Engulfing. The capsule is a major virulence factor. Know that. We have so many virulence factors, but capsule is a major virulence factor. I hope you know what they call virulence. Pathogenicity and virulence, they are related. So, you know, so like I told you, we have capsulated bacteria and we have non-capsulated bacteria. Not all of them are capsulated. So another structure of bacteria is cell wall. This cell wall provides rigidity for the cell and is composed of peptidoglycan. The wall, that is cell wall, give the cell its shape and surround the cytoplasmic membrane. So the function of the cell wall is to provide rigidity and give the cell its shape. It also helps to anchor appendages such as pili and flagella. So it's the one that fasten the pili and flagella onto the cell wall. So then we have cytoplasmic membrane. It's a layer of phospholipid and protein. It is also called plasma membrane. It's either you call it cytoplasmic membrane or plasma membrane or even cell membrane. So when you say cell membrane, you are referring to cytoplasmic membrane. When you say plasma membrane, you are referring to cytoplasmic. So this plasma membrane encloses the interior of the bacterium, regulating the flow of material in and out of the cell. So cytoplasmic membrane, it regulates the in and out of material That is what it does. So flagella, singular flagellum, they are whip-like structure that provide means of locomotion. So the main purpose of flagella is for locomotion, for movement. We have pili. Pilus is the singular, pili is plural. They are hair-like projection emerging from outside the cell surface. So what do they do? What is their function? They assist bacteria in attaching to other cells. So through pili, bacteria can mate with another bacteria through conjugation. Are you there? So they assist the bacteria in attaching to other cells and surfaces. Without pili, many disease-causing bacteria lose their ability to infect because they will need to cross over to the other one before they can infect the host of any or another bacteria. Then ribosome, they are microscopic factories found in all cells, including bacteria. They translate the genetic code from molecular language on nucleic acid to that of amino acid. That's the function of ribosome. Translation of genetic code to amino acid, which is the building block of protein. Amino acid is the building blocks of protein. So how do bacteria, how do they reproduce and grow? They can reproduce through binary fission. And this is an asexual process. That is, it doesn't need two bacteria before they can divide. So binary fission. In this process, the chromosomal DNA duplicates, after which bacterial membrane and cell wall grow. There is what we call budding. This is another form of asexual reproduction, in which new organism develops from an outgrowth or bud due to cell division at a particular time. Then we have fragmentation. Some filamentous bacteria and fungi reproduce by the mode of asexual reproduction, where a parent organism breaks into fragments, each capable of growing into a new individual. Then we have growth phase. We are used to growth phase. They have been teaching us growth phase since our techo days, or through our part 1 or part 2. So we have lag phase, have logarithm phase or log phase, we have stationary phase, and we have death phase or decline phase. All these things, I don't need to mention it because we all know what is called lag phase, log phase, stationary phase, and decline. What's the time?
4 minutes to 9.
1 minute to 9.
4 minutes.
4 minutes to 9,
4
sir.
minutes to 9. Because I don't want us to. Because I know some people are just, they didn't want to talk the way they are. Don't know. Maybe they are cursing me now that they want to sleep. Okay. Last thing that we will touch here, we did it at 400 level, but let me just quickly go through it because without it, there won't be any microbiology. Sterilization and disinfection. We have different methods. We have physical method, which include the following: heat sterilization. The heat can be dry heat and it can be moist heat. When you say moist heat, that is you are using autoclave, that is steam under pressure. The principle is steam under pressure. And at the end of the day, there will be coagulation of proteins in the microorganism and the organism will die. Then the dry heat makes use of incinerator, hot air oven, and flaming technique. So this one, the principle is oxidative damage. And once there is oxidative damage, the bacteria will die. Then there's another one under the physical. Filtration. This method uses membrane filter. This one uses filter. Just filter it, and some of the organisms may be able to be filtered out. Irradiation. All these things we did it at 400 level. I'm just trying to go through it for you because there's no way you will do microbiology that you will not be doing sterilization. Irradiation is a process of exposing surfaces of objects to different sterilization. We have different types. Non-ionizing radiation, that is the use of ultraviolet radiation. Then ionizing radiation, such as gamma rays and X-ray. We have talked about physical. Thank you. Then the chemical methods. This method involves the use of chemicals to sterilize surfaces or objects sensitive to any object that is sensitive to high temperature. You don't use either dry heat or moist heat. You use chemical method. So we have gaseous sterilization, which involves the process of exposing equipment or device to sterilizing gases in a closed, heated, or pressurized chamber. We have different types of these sterilizing gases. Examples are ethylene oxide, formaldehyde, nitrogen dioxide, and ozone. Then there's another one, liquid. You know, under chemical, we have talked about gaseous sterilization. There's also liquid sterilization, which involves submerging of equipment in a liquid sterilant to eliminate all viable microorganisms and their spores. Common examples of this are hydrogen peroxide, glutaraldehyde, and hypochlorite solution. So you should be able to know some example of this because it can come out in your whole business. So disinfection, what do we mean? What do we call, what do we understand by disinfection? Disinfection is defined as a complete elimination of microorganisms except bacterial spores. You can see except. You can eliminate microorganisms, but if it is one that has spores, it won't be able to eliminate them. The primary goal of disinfection is to reduce the risk of infection by pathogens. The types of disinfectants include what we use for what we call disinfectant in laboratory. Example of them is aqueous, especially ethanol and isopropanol. We have aldehyde. Example of that one is glutaraldehyde and formaldehyde. We have oxidizing agents, hydrogen peroxide and peracetic acid. We have halogens. These are chlorine compounds, e.g. sodium hypochlorite. They are example of halogens. They are chlorine compounds. And under this, we have also iodine compound. And all these chlorine compound and iodine compound, they are called halogens, and they are used for disinfection. Then there's another quaternary ammonium compound. These compounds are effective against bacteria, fungi, and envelope viruses. They are used for surface disinfection in healthcare settings. Then, last but not the least, is phenolics. Phenolics and its derivatives are used for disinfecting surfaces and equipment. They are effective against bacteria and fungi, but have limited activities against viruses and spores. This is convenient place for us to stop today. Thank you for listening.
Thank you, sir.
Are we all there?
Yes, sir.
Yes, sir.
Yes, sir, but I didn't hear the last one. You said the last, but not the least. I didn't hear it.
Phenolics,
Phenolics.
Phenolics, okay.
phenolics, and they are phenol and its derivatives. Phenols and its derivatives are called phenolics.
Okay. Thank
So
you.
the phenolics have limited activities against viruses and spores. So these are
Okay.
the drawbacks. So that so we can just ask you to write on disinfection. So these are the things that
Thank you, sir.
you can put down under disinfection. So be able to show that you followed the class. So far so good. I hope I've not taken much of your time tonight. I had told myself that I don't want to creep into your relaxation time. I won't spend more than 1 hour, and I hope I did well with time. So, any
Yes.
question?
Yes, sir.
I hope you are happy that this
Yeah,
man
thank
did
you,
not
sir.
go on 2 hours lecture today, 3 hours.
Yeah, well done, Dr. Akinabi.
We have gotten to yeah. We
Can
have
we have
gotten
the
to 5 minutes.
slides of
You
tonight's
say
lecture?
you say
I
what?
said, can we have the slide of tonight's lecture?
Definitely, you will have the slide. You will have the slide.
Okay.
So we'll send the slide through the platform. So maybe our next lecture will be able to go into, I think, host parasite relationships. After that, I think we'll now be going into individual bacteria. So I promise that lecture will not be lengthy again. We are in 500 level, and most of the things we already know. I've dropped all those things that are not important to us as a microbiologist. The cytology has gone this way. The topology has gone their way. The chem part has gone this way. So we are focusing on microorganisms. Thank you. Any questions before we call it a day? Are we all there?
Yes, sir.
Yes, yes, sir.
Any question? And if there's no question, let me go and do other things.
Okay,
Okay,
good night,
sir.
sir.
Thank you very much, sir.
Good night,
Yes,
sir.
sir.
Good night. And
Good
Good
I
night,
will
sir.
let
night, sir.
Thank
the
you,
class
sir.
Yeah.
welcome my sisters and brothers. I will let you know the next time I'm having class with you.
All right,
So
sir.
Thank you,
nothing is
sir.
cast, nothing is cast on a stone. It's flexible. I will continue to be flexible in our approach. So thank you.
Thank you, sir. Good night, sir.
Good night.
Thank
Good
you.
night. Thank you, sir.
Good night.
Good night, sir.
Mhm.